Hello and welcome back to educator.com. Today we're going to talk about translation. So as an overview, first thing we need to talk about is how we can link transcription to translation. After that, we will just give an overview of translation and go into it all the way through. So first we'll talk about ribosomes and their ribosomal subunits, and then we'll go through each part of translation, so starting with the charging of tRNA, all the way through to initiation, elongation, and termination of translation. And finally, we'll briefly talk about the consequences of altering the genetic code. So transcription, once again, to remind us from last time, is making RNA from DNA. Okay, so we synthesize RNA, okay, complementary to DNA. All right, and this is going to occur in the nucleus. All right, this is important to remember. Okay. Now, translation is the process of synthesizing a polypeptide from an mRNA transcript. All right, so synthesize polypeptide from mRNA transcript. Now, if we have our tRNAs, our rRNAs, those aren't going to be made into proteins. So you aren't going to translate those RNAs. All right, and obviously our non-coding RNAs you won't translate either. But our mRNA transcripts are the ones that are going to go through translation very often. All right, so we have our mRNA being translated in a 5' to 3' direction, all right, just as if we were talking about DNA synthesis happening in a 5' to 3', meaning the next base is added to the 3' end. Okay, so mRNA is translated 5' to 3', okay, 3 bases at a time. All right, this is important to know. All right, 3 bases at a time, which we call a codon. All right. So each 3 base pair unit, or each 3 base unit on an mRNA is called a codon. And each one of those codons will code for a specific amino acid or a stop. All right? So the mRNA gets translated 3 bases at a time by base pairing with a tRNA, okay, having a complementary anticodon. So your codon and your anticodon will match up via a base pair. So an example being, let's say, UAG on your codon, your mRNA, okay, would match up with AUC on your tRNA. All right, and remember, we would have the hydrogen bonds between there, right? Okay. Now, translation is going to occur in the cytoplasm. And it will either occur inside ribosomes that are located on the rough endoplasmic reticulum, okay, which the endoplasmic reticulum is studded with ribosomes. That's why it's called the rough endoplasmic reticulum. We also have the smooth ER. So it's going to be either in the rough ER or in the cytosol itself. All right. And so if we look at this depiction down here, we can see that here is our nucleus. All right. We have transcription occurring in the nucleus, so DNA being made into RNA. In this case, we're looking at mRNA. That mRNA can leave the nucleus into the cytoplasm, at which point right here, this is our ribosome. Okay, and it's going in this direction. Our ribosome can be the site of protein synthesis by reading the mRNA 1 codon at a time and recruiting our tRNAs to come in and add its individual amino acid to the growing polypeptide chain shown here. Okay, and we'll go over each one of these steps of translation today, and hopefully we'll have a very good understanding by the end of this unit. So, as an overview, what's happening in translation? All right, so first things first. We have a ribosome binding to an mRNA molecule. All right, it's going to bind to it and it's going to search for the start codon. Okay, now a start codon, this is going to be found once again on the mRNA. Your start codon is going to be AUG. And remember, this is on the mRNA. Okay, this AUG will code for methionine. Okay, methionine. Now, inside the ribosome, charged tRNAs, so that means they're carrying an amino acid, will base pair to the mRNA via the complementarity found between the anticodon on the tRNA and the codon on the mRNA. Okay. So this is important. Codon on the mRNA, anticodon on the tRNA. Okay. Now the next step is we have the amino acids being transferred from the tRNA and being linked to the growing polypeptide chain via a peptide bond, otherwise known as an amide bond. All right? And this growing polypeptide is still being associated with the ribosome itself. Okay. Now, spent tRNAs, so tRNAs that no longer have an amino acid because it's already given its amino acid and let it attach to the growing polypeptide chain. These spent tRNAs get released from the ribosome via the E site, okay, or the exit site, and we'll talk about that later. And then the process of bringing in a new tRNA, having a peptide bond being synthesized and a growing polypeptide continuing in its synthesis will continue until you reach a stop codon. And once again, remember it's a codon, so it'll be found on the mRNA. And our stop codons are UAA, UGA, and UAG. Okay, so we continue till we reach a stop codon, and then what will happen is instead of a tRNA coming in, we have a protein called a release factor coming in, and what that does is it basically hydrolyzes or cleaves the polypeptide, releasing it from the ribosome and allowing it to fold into its final tertiary shape. Okay. So those are our main steps. That's our overview. So let's first start with what our ribosomes are and what the subunits are. So first of all, a ribosome is it's a nucleoprotein. Okay, so it's made up of several proteins as well as ribosomal rRNAs. So remember, our rRNAs plus many proteins will make up our ribosome. Okay, so we have different ribosomes in eukaryotes and prokaryotes, but they serve basically the same function to synthesize proteins. So let's first look at our prokaryotes. Okay, our prokaryotes, the overall ribosome, okay, it's the same thing as if we were looking down here, okay, is a 70S. And what that is, an S is a density unit, okay, and it's a Svedberg unit. Okay. Now, we have the small subunit, which would be down here, being a 30S small subunit, and the large subunit, which is the top part, the bigger part, would be the 50S subunit. All right, and so in our prokaryotes, the rRNA that is included in the 30S, or the small subunit, is our 16S rRNA, as well as several proteins. And then our 50S contains the 5S and 23S rRNA, as well as our protein subunits. Now, what's important, we have this 23S rRNA, and this is our ribozyme. Okay, so it's our peptidyltransferase. All right, so that's our peptidyltransferase. In eukaryotes, we have what's called an ADS ribosome. Okay, so it's a little more dense, and we have our small subunit, including our 18S rRNA as well as proteins, and that's called the 40S subunit. We have our large subunit being the 60S subunit, and that contains 3 different rRNAs, our 5S, our 58S, and our 28S rRNA. And our 28S rRNA is also the ribozyme found in the eukaryotic one. So that's the peptidyltransferase enzyme in eukaryotes. All right. And now each ribosome, fully functional, okay, full unit ribosome, our 70S and 80S, each complete ribosome has 3 different very important sites. We have the A site, okay? The A site, which can be found right here, is known as the aminoacyl site or the acceptor site. We have the P site, which is known as the peptidyl site, the peptidyl tRNA site. This is where we're actually making the new peptide bond. The A site is where the new tRNA will come in. And then we have the E site, which is the empty or exit site, and that would be, I guess it's not on here, but this would be the E site right here, and that's where the spent tRNAs are leaving the ribosome, so they're being released. Okay. So let's talk about our major steps of translation again. So first of all, we have the charging of the tRNA, followed by initiation, elongation, and termination. So in activation or charging, the proper amino acid is esterified to the tRNA by its carboxyl group, okay, to the 3' OH of the tRNA. So remember, a bond between a carboxylic acid and an alcohol, okay, makes an ester. All right, and if we remember, right, our tRNA, right, our cloverleaf structure, we have this CCA at the end, okay, at the 3' end, and that A is where we are going to esterify the amino acid. So let's say, for example, this would be methionine. Okay, so methionine would be added right there. Okay. Next, we would have initiation. All right, so initiation would involve the small subunit of the ribosome binding to the 5' end of mRNA with the help of initiation factors, okay, which we'll abbreviate as IF. Now, the next step would be elongation, and that's going to happen when we have the next aminoacyl tRNA. So this is the charged tRNA. Okay. When the next one comes in, binds to the ribosome along with GTP, because this is very energy. You need a lot of energy for this to occur. All right. So when we have energy and a charged tRNA coming in, as well as an elongation factor, we can allow elongation to occur. So we're lengthening the polypeptide longer and longer. Finally, we have termination occurring when we have the acceptor site of the ribosome encountering a stop codon on the mRNA. And remember, our stop codons are UAA, UGA, and UAA, UAG, and UGA. So when this happens, we have a releasing factor. If it's eukaryotes, it's ERF1. If it's prokaryotes, it's just RF1 or RF2. And that will cause the release of the polypeptide chain from the ribosome because it's hydrolyzing it and basically releasing it. Okay. Now we'll get more into the release factors as we hit the termination section in this presentation. So let's start with the first step, the charging of our tRNA. So we have an enzyme called aminoacyl tRNA synthetase. Okay, and that is going to catalyze a 2-step reaction that results in the covalent attachment of the carboxyl group of an amino acid to the ribose of the adenosine, okay, of the CCA. Let's see right here. Okay, this is, let's say, CCA. All right, so we have the carboxyl group of an amino acid. Okay, let's say we have methionine. Okay, and we're gonna say it's carboxyl group. Okay, so its carboxyl group is going to attach via an ester bond to the adenosine on this 3' end. Okay, so this is 3'. This would be 5'. And once again, this is a tRNA. Okay, so we have an ester bond all right. So we have our covalent attachment. All right, right here, it's an ester bond between the ribose of the adenosine found on the 3' end of the tRNA and the carboxyl group of the, in this case, methionine, but it can be any amino acid. Okay. So how is this occurring? We have it's a two-step reaction. We're not going to get into all the detail, but we have that esterification. We have two different types of aminoacyl tRNA synthetase enzymes. We have class 1, and that attaches the amino acid to the 2' hydroxyl group of the ribose. We have class 2 that attaches the amino acid to the 3' hydroxyl of the ribose. So the end result is the same. We have an ester bond between our tRNA and the amino acid. It's just where it is occurring. Okay. Now, what's very important about this reaction is that it is highly specific. Okay, and the reason that it is highly specific is because, let's say this anticodon, okay, it has to be specific, hopefully, for the methionine. So this would be specific for the methionine, all right, whatever that 3 base pair sequence would be. Okay, if for some reason we have a different amino acid, the methionine, being attached to this specific tRNA, what will happen is when this tRNA properly base pairs with our mRNA, which has the codon for the methionine, okay, so this is supposed to hydrogen bond together. If this amino acid is not methionine, we are going to add the improper amino acid to the growing polypeptide chain, even though we have proper base pairing between the codon and anticodon. Now, the whole reason that this is a highly specific reaction is that we have high fidelity of our translation, because if we start adding in amino acids that aren't supposed to be attached to the specific tRNA according to the anticodon, then we are going to mess up that protein and could really, really detrimentally hurt that cell. Okay. And the last thing I want you to know about our charging is that we have ATP energy being required. All right. So once we've charged our tRNA, we can continue on with initiation. So let's start with our prokaryotes, right? Our bacterial and virus section. So let's look at it. So first of all, we have initiation factor 3 binding the small subunit, the 30S, at the E site, the exit site. The whole reason that it does this is so that the 50S subunit cannot bind. Okay, so if we have the exit site here, we have IF3 binding, and that does not allow, okay, it does not allow the full 70S ribosomal, the full 70S ribosome complex to be constructed. All right. And so the IF3 protein binds the 30S subunit after the previous round of translation is finished so that you can't rebind. Next, we have IF1 binding the 30S subunit at the A site. Okay, so this would be the P, that would be the A. Okay, so we have IF1. Okay, and that prevents any tRNAs from binding because remember, the tRNAs are going to come in at the A site. Okay. Next, we have IF2 and GTP binding IF1. And then, finally, we have our 30S, the small subunit. Okay, let's draw that. So this is the 50S. This is the 30S. Our 30S subunit will then associate with the mRNA. And it'll do that by binding to what's called the Shine-Dalgarno sequence, which has a consensus sequence of AGG, AGG. And it's near the 5' end of the mRNA. And so the mRNA, let's draw it in purple. The mRNA will bind the mRNA will bind and to the 30S subunit using its Shine-Dalgarno sequence, okay? And the Shine-Dalgarno sequence has homology to the 16S rRNA that is found in the 30S subunit. So we have the 16S rRNA. Okay. Now, once that happens, we have a special tRNA coming in. So we have a tRNA that's carrying an N-formylated methionine. Okay? And by the way, guys, what a formal group is just a C double bond O H. When it's bound to an R group, we just call it an aldehyde, okay? All right, so our informal, so it's a formylated methionine, so it has this group attached to it, binds to the mRNA at the proper start site, the AUG. Okay, that is facilitated by our IF2 and GTP that's already there, and that helps position the start codon in the P site. Okay, the P site is where we are going to have the peptidyl transferase reaction. So that's where we're actually going to catalyze the synthesis of that peptide or amide bond. Okay, so at this point, once we have this tRNA, this formylated methionine tRNA bound in there, we have a complete 30S initiation complex. All right. After this occurs, IF3 will be released, letting now the 50S subunit bind. Remember, IF3 was the one that didn't allow the 50S subunit to bind after the previous round of translation. Okay. What happens next? We have IF1 and IF2 being released, and now we have a fully formed 70S initiation complex that has both an empty A site and an empty E site, and we have this N-formylated methionine tRNA in the P site. Okay. Now, this formylated methionine, we don't have to worry too much about because we have an enzyme called deformylase that removes this formal group. Okay. from that met tRNA either during or after translation. So we don't see the formulated version in the mature protein. Okay. All right, so let's draw out these prokaryotic initiation complexes. So first off, let's draw out the 30S initiation complex. Okay, so first off, we have the 30S ribosomal subunit. Okay, we have the E site, the P site, the A site. Okay, we are going to have our IF3. Our IF3 protein binding the E site, and that makes sure that the 50S subunit cannot associate with the 30S. Okay. We then have IF1 binding the A site. We also have IF2 and GTP binding IF1. Okay. We will then have the mRNA. Okay. The mRNA coming in, binding the ribosome via homology to the 16S rRNA. Okay, and that the 16S rRNA is binding to the Shine-Delgarno sequence found on the mRNA. And finally, we will have that formulated tRNA, okay, coming in and binding to the P site. So we will say and this is we'll just call it MET I for the initiation. Okay, so it's the formulated one. It's a special one. Okay, so that's our 30S initiation complex. Now let's look at our 70S initiation complex. Remember, S is a Svedberg, right? It's a density unit. Okay, so what we have now is our 30S. Okay, it still has its exit site, its peptidyl site, and its acceptor site. We have that mRNA, okay, properly in there. Okay, we have lost IF3, so now we can have our 50S subunit binding. IF1 and IF2 have left as well, and we still have our original tRNA with that initiation special methionine in there. Okay, and remember, the anticodon of the tRNA is going to base pair complementary with the codon on the mRNA. All right, so this is our 70S initiation complex. So what about translation initiation in eukaryotes? Same concept, a little more complicated. Okay, so in eukaryotes, the small subunit, our 40S, is actually already associated with the initiation tRNA before it's recruited to the mRNA. All right. And then what we do, okay, we will form what's called a 43S pre-initiation complex. And what that consists of is the following. We have the 40S subunit, the ribosomal subunit, okay, and that's going to bind to the 5' cap of the eukaryotic mRNA. Remember, the eukaryotic mRNA has the 5' cap, all right? Now, what we have after the 40S subunit binding to the cap, we have eukaryotic initiation factor 1 binding to the E site. We have EIF5 binding to EIF1, EIF1A binding to the A site, EIF3 binding to that one, EIF1A, and then EIF2 and GTP binding the initiator methionine tRNA and recruiting that to the peptidyl site, the P site. Okay. All right. Now, part of eukaryotic translation initiation is the circularization of the mRNA. And this occurs via a complex called EIF4, and it's EIF4A, EIF4G, EIF4E. So EIF4A will bind to the upstream region of mRNA. And let's draw this out as we're talking about it. So first off, we have the mRNA that we're going to draw. Okay? We will denote that it has a cap right here. Okay? So EIF4A will bind to an upstream region on mRNA. So let's say here is A. Okay? EIF4G binds to the poly A tail, okay, via hydrogen bonding. Okay. And EIF4E binds to the 5' cap. Okay. Now, what this allows is for, let's say, we'll say where the start codon is. Okay, so remember, this is all upstream. So let's say the start codon is right there. Okay, so here's your start. And remember, our start codon is AUG, and AUG codes for methionine. Okay, so what we can have by circularizing this, we actually allow ribosomes, let's use a different color. We allow ribosomes, we'll use green. Ribosomes are allowed to bind in several different places. Okay, so small subunit, large subunit. Okay, all the way up until, let's say, here's our stop. Our stop codon. And remember, our stop codons are UAG, UGA, or UGG. Okay. Okay. Oh, sorry, not UGG, UAA. Sorry about that. UAA. Okay, UAG, UGA, UAA. And these, remember, do not code for a tRNA, and therefore do not code for an amino acid. They code for a release factor. So each of these ribosomes, okay, can bind to this circularized mRNA, okay, making what we call a polysome. And what we'll have is a little bit of protein, right? And as it goes further along, it'll get longer and longer. Okay, these are all different ones. Okay, so these are all proteins. Okay, those are all being made, being synthesized, so translation is occurring at that time. So what we're doing is we're making several different proteins, or probably the exact same protein, but we're making several copies of the protein from a single mRNA. Okay, so if we have multiple ribosomes translating, we're getting multiple copies of the protein from a single mRNA. All right. Okay, so we talked about the 43S initiation complex. Now let's move to the 48S pre-initiation complex. Okay, so the 48S pre-initiation complex is the 43S that we talked about plus the addition of the circularized mRNA with the the EIF4, EG, and A subunits. Okay, so the 48S pre-initiation complex will scan the mRNA 5' to 3', looking for the AUG with the correct reading frame. So the AUG, meaning it's looking for the start codon, the methionine, okay, and it's looking for it in the correct reading frame. At the very end of this unit, we'll talk about reading frames. So for right now, let's just move on a little bit. So this right here, looking for the AUG in the reading frame, is an ATP-dependent process, so it's energy using. Okay, and it's facilitated by our EIF4A, which actually has helicase activity, and EIF4B. EIF4B actually activates EIF4A. All right. So once the proper AUG is found, our initiation factors 1, 2, and 5 release, which allows EIF4B and GTP to bind that initiator tRNA. Okay, so we lose a bunch of initiation factors and gain one which helps a tRNA bind. Okay, this then recruits the large subunit to associate, making the fully functional ADS ribosome with the proper initiator methionine tRNA loaded into the peptidyl site. So similar to the the prokaryotic initiation, we have all the initiation factors, get everything ready, then they leave so that you can have the full association of the large and small subunits with the proper initiator tRNA in the peptidyl site. Okay. And just as a reminder, we have the cap, or if we don't have a cap, which is actually fairly common on mRNAs and eukaryotes, if you don't have the cap, you utilize what's called a Kozak sequence with the consensus sequence ACC-AUGG. And whether it's the cap or the Kozak sequence, that will help the 40S ribosomal subunit bind to the mRNA. And then the 60S subunit will bind at the AUG, as we talked about up here, once it's been found. So let's show what this would look like. So we have our 43S pre-initiation complex. Okay, so here we go. We have our 40S ribosomal subunit. Okay, with the E, the P, and the A site. All right. Now, we have EIF1 binding here. Okay, and this does not allow the 60S subunit to bind. All right. We have EIF 5 binding this. Okay. Over here at the A site, we have EIF 1A binding here. That's blocking the acceptor site so that we can't have new tRNAs coming in. And then we have EIF3 binding that. Okay. Now we have our tRNA coming in here. Oh, let's use a different color. Okay, and this is going to be attached to that specific initiation methionine. And then we have, oh yes, our last one, we have EIF2. We have EIF2 plus GTP. Okay, binding here. Okay, and that's going to help position the methionine in the proper P site. Okay, so this is the 43S. All right, now if we talk about our mRNA, let's just say here we have our mRNA, our cap here. Okay, I'm going to draw it in the non-circularized form just so we can see it easier. So we have EIF4E bound to the cap. Okay, we have EIF4G bound. We have the EIF4A helicase, and then we have EIF4B, which activates this helicase, bound right here. So this, okay, this one plus this one equals our 48S pre-initiation complex. Okay, and just so we can see one more, let's write out what happens when we have the full association. So we have our 40S. We have our E, our P, our A. Once we have our initiation factors leaving, that allows us to have association with the large subunit, okay, the 60S. And then we have that mRNA coming through, right? And we have our proper initiation tRNA in the P site. We will have an empty E site and an empty A site, and then we now have the full 80S complex. And now it is ready to go through elongation. Okay. Okay. So the next step, elongation. So now this is going to just repeat over and over again. Okay, elongation. Every time a new tRNA comes in, this is a step that's going. Okay, so we have charging happening to every tRNA. We have initiation happening once. We have elongation happening for every tRNA that's coming in, and then we have termination happening once. All right, so we have our incoming charged tRNA, right? Aminoacyl tRNA being recruited by, if we're eukaryotes, it's EF1 alpha, so elongation factor 1 alpha, or for prokaryotes, elongation factor TU. All right, so the incoming charged tRNA binds the complementary codon on the mRNA in the A site. Okay, and remember, it's using its anticodon to bind the codon. Next, we have peptide bond formation being catalyzed by the enzyme peptidyl transferase. All right, and remember, that is a ribozyme, either the 23S rRNA, if it's prokaryotes, or the 28S rRNA, if it's eukaryotes. Okay, so the peptide bond formation occurs between the amino acid of the tRNA that's in the A site, so it just came in, and the C-terminal amino acid of the growing polypeptide chain, which is attached to the tRNA that's already in the P site. Okay, and I'll draw this out in just a couple slides. And finally, the growing polypeptide chain gets transferred to the tRNA in the A site, and we will have what's called translocation occurring. So, as I said, translocation occurs, and that is enabled by the action of elongation factor 2 if it's eukaryote, EFG if it's prokaryotes. So translocation moves the tRNA in the P site. Now it doesn't have an amino acid anymore. It moves it to the E site. All right. So remember, the polypeptide chain has been transferred already from the tRNA in the P site to the tRNA in the A site, and now we do a shift. Okay. So the tRNA that was in that A site, so the one that had just came in, now is attached to the growing polypeptide chain. Will move to the P site. All right. And now the tRNA that was in the E site, the exit site, is released, and the next charged amino acid, which is dictated by the codons on the mRNA, will enter into the A site. So let's draw this out so that we know what it looks like. All right, so I will draw my ribosomes in blue. Okay, and I'll label this once for us. We have 40S and we have 60S. Okay, we have the E site, the P site, the A site. All right. Oh. Okay, this is our mRNA. And it's the 5' end. Okay? And we're going to need to draw this bigger. Okay. So let's make the... Okay. So we have the E, the P, the A, right? And we're going to have the same channels in the 60S as well. Okay. Our mRNA. This is 5' mRNA. So at first we have, I'm just going to very simplify what a tRNA looks like. All right, so I'm just going to draw it just kind of like this. Okay, attached to, let's say, a particular amino acid. Okay, so that's what I'm going to say an amino acid looks like. Okay, so this is perfectly what we have for initiation. Okay, so the next step, as the mRNA, okay, kind of stays in one place, the ribosome will actually shift to the right. Okay, so we'll now go to the next codon. What we have is, or what the next step would be a a new amino acid, or sorry, a new charged tRNA coming in with a different amino acid, let's say. Okay. Good. Now, what we have is called translocation. Okay. And what we have (60S), (40S), we have EPA, EPA. We have, as the ribosome moves, okay, what we end up having is kind of a shift looking like this, whereas the tRNAs are still bound in the P and A site. However, they've shifted looking like this, and what we're actually doing in that P site, we are what we're doing, okay, it goes red, then blue. We are making that peptide bond. So if I move it, okay, what we've done is we've transferred this amino acid, right, the methionine, to this, right, to this tRNA. So we've made a peptide bond or an amide bond. Okay, and now we have a growing chain. As we can see, the first amino acid was the methionine, the blue. The second amino acid could be whatever you want to call it. Let's just say it's alanine. Okay, and we've had that translocation. Now, the next step would be the full movement to where it looks like this. (40S), (60S), the EPA, EPA, okay, the 5' mRNA. Now, we have a full movement as we're translocating, right? So now we have okay, so now we have the first tRNA, now without an amino acid attached to it, is moved into the E site. The last the last tRNA that has come in through the A site now has 2 amino acids attached to it. Okay, so the growing polypeptide chain is now fully in the P site. And now that opens up the A site for the next tRNA to come in. So let's see what that looks like if we continue this way. Okay, so what we have here is, okay, so it's gone out already. We now have this one here. Okay, so that's exited. And now we have a new tRNA coming in with another amino acid. and the whole thing will occur all over again. So now you can just look, what would happen next would be, this would be the next step, okay? And it would follow to here and then back to here again. Okay, so that would continue and over and over again until we hit a stop codon, in which case then we will go through termination. All right. So now that we're talking about termination, we need to talk about release factors. So these are proteins that will terminate translation when the ribosome comes to a stop codon. Remember, stop codons, UAA, UAG, UGA. All right, so the release factors are proteins. They're not tRNAs. They don't carry an amino acid. This is very important to understand. Okay? So important that I'm just going to highlight it all. Okay? And I'm even going to put a red star by it. Okay? All right. Now, there are two classes of release factors. We have class 1 release factors. They will recognize stop codons and hydrolyze or cleave the polypeptide chain from the tRNA that is found in the P site. Okay? We have, if we're talking about prokaryotes, okay, so these are prokaryotic release factors. Release factor 1 recognizes UAA and UAG. Release factor 2 recognizes UAA and UGA. All right? Eukaryotic release factor 1 will recognize all 3 stop codons. Okay? Now, class 2 release factors will help release class 1 release factors from the ribosome. So, class 1 release factors cleave the chain from the tRNA in the P site, so it allows the protein to leave the ribosome. Class 2 release factors release the class 1 release factors from the ribosome. Okay, and the class 2 release factors are RF3 in prokaryotes or ERF3 in eukaryotes. So I put together a little table so that it might be easier for you guys to understand and have all the information in one place. So this should just say step. Okay, so your tRNA charging, the whole function of that is adding an amino acid to a tRNA, and the protein or the enzyme that's responsible for that, okay, is aminoacyl tRNA synthetase. Now, one thing I want to point out, guys, synthetase, a general rule of thumb, when you see the word synthetase, that means it's an enzyme that's making something, so synthesizing, and it needs the input of energy. Usually ATP or GTP is needed, okay? If this were synthase, okay, it's an enzyme that's doing a synthesis reaction but doesn't need the input of energy. All right. That's just kind of a biochemical aside. All right, so you would learn that in biochemistry. So the next step of translation is initiation. So the recognition of the start site for eukaryotes, that's done by EIF1A and EIF3. For prokaryotes, that's IAF3. Recruiting the special initiator methionine tRNA to the P site. Eukaryotes, that's EIF2. Prokaryotes, that's IF1 and IF2. Okay. For elongation, recruiting all other tRNAs to the A site. Okay, prokaryotes, that's EF1 alpha. For prokaryotes, EF2, as in TU. For peptide bond formation, whether it's eukaryotic or prokaryotic, the name of that enzyme is peptidyltransferase, and it is a ribozyme, okay, meaning it is a ribonucleic acid that has enzymatic activity. For eukaryotes, that's the 28S rRNA, and prokaryotes, that's the 23S rRNA. Okay, and either one of those are found in the large subunit. So eukaryotes, that 28S rRNA is found in the 60S subunit. Prokaryotes, the 23S rRNA is found in the 50S subunit. Okay, and finally, translocation involved in elongation. That is EF2 for eukaryotes, EFG for prokaryotes, and you're going to need GTP for each time it translocates. So it's energy requiring. And finally, termination. This is the recognition of stop codons and the release of the polypeptide chain. That is done with our class 1. The hydrolyzation of the polypeptide chain from the tRNA in the P site is done by class 1 release factors, which is ERF1, or if we're talking prokaryotes, RF1, RF2. And then the class 2 release factors, which release the class 1 release factors from the ribosomes, is ERF3 for eukaryotes and RF3 for prokaryotes. So let's do a quick review of translation. So each tRNA with its own anticodon is attached to a specific amino acid, right, via the CCA end on the 3'. The amino acids delivered by the tRNAs are joined together to form a polypeptide inside the ribosome. And remember, the tRNAs utilize the complementarity of its anticodon to the codon on the mRNA. The ribosome will move along the mRNA and encounter codons, which are 3 base sequences that will tell whatever tRNA to come in and add its specific amino acid. So multiple ribosomes can translate the same mRNA at the same time, creating a polysome, which I talked about when I circularized the mRNA. The next thing that happens, we have a charged tRNA entering the A site, where the anticodon hydrogen bonds to the codon. Next, the tRNA translocates to the P site. Okay. The attached amino acid gets connected to the previous amino acid of the growing polypeptide chain. And then the spent tRNA, the one that no longer has an amino acid on it, exits from the E site. And this continues over and over and over again until a stop codon is reached. Now, I briefly said that I wanted to talk about consequences of altering the genetic code. And so we have just a couple slides, and I want to explain to you why this is so important. So our genetic code, right? If we look back at our codon table, which I have on the next slide, each 3 base sequence, which we call a codon, codes for a specific amino acid. And each one of those amino acids are in a protein, okay, for a specific reason, right? So maybe they're supposed to make hydrophobic bonds or hydrophilic bonds, ionic bonds, so on and so forth. So if you alter that sequence, so if you're adding in an amino acid that shouldn't be there, you can affect the overall function of the protein. So there are a couple different types of mutations that I want to talk about. We have first what are called silent mutations. These are the types of mutations that we can call no harm, no foul. All right? So these are mutations in the DNA sequence that do not alter the amino acid sequence, okay, due to our degeneracy of the genetic code. So example, all 4 of these, and actually two other triplets, but all 4 of these code for serine. So if we happen to, instead of what should have been a UCU, if for some reason we have a mutation changing the U to a C or even an A or a G, no harm, no foul. We still get a serine. Okay, so it's a silent mutation. We don't even see it. Now, if we get what's called a missense mutation, that's a mutation in the DNA sequence that leads to a change in the predicted amino acid sequence. So, for example, instead of a UGC that's supposed to be added, okay, there's some sort of mutation, and we actually have a G present instead of a C in that 3rd triplet base. That changes the cysteine to a tryptophan. So maybe we were supposed to be making a disulfide bond using our cysteines, in the protein, but now we no longer have that ability. So that can be detrimental. Now, missense mutations don't always have to be problematic, because let's say we have aspartate and it is changed to a glutamate. Well, you still have a negatively charged amino acid, and maybe it can participate in the same exact bond that it was supposed to have made. So missense mutations don't always have to be showing a type of phenotype, but they can. Right? Now, the last one, the most detrimental one, is a nonsense mutation. So that's a mutation in the DNA that changes a normal amino acid coding codon to a stop codon. And this truncates the protein, so it makes it smaller. So an example, we have UAU coding for tyrosine. Okay, but all we have to do is have that one base change to UAA being a stop codon. So maybe instead of getting a full, let's say, 6,000 amino acid protein, maybe we only get 60 amino acid protein. And if that's the case, it's not going to be very functional, and we're likely going to degrade it and just throw it away. What can cause even more problems is if, let's say, we get a protein that can still function, but it functions in a different way, and thereby maybe causing a genetic defect. Okay. So remember that the consequence of altering the genetic code are due to the possibility of changing the triplet code, right? So cysteine, it's fine if you change that last one, the U, to a C, right? Not a big problem. But if you change that U or C to an A or a G, to an A, that's going to give you a stop codon. So that's an example of a nonsense, right? If you change the U to a C right here, that's an example of a silent, because it's still going to give you cysteine. If you change the U to a G, that's going to give you a different protein, right? Or a different amino acid, and that's an example of a missense. So between these two, silent. Between these two, nonsense. Between these two, missense. Okay. So I want to end with just an example of what a frame shift mutation is, okay? Because this can alter the genetic code. Now, frame shift mutation is due to the addition or deletion of nucleotides in other than multiples of 3. Okay, and that will shift the open reading frame, which most often results in truncated proteins. So let's write out a sequence. So A, C, G, right? And this is the mRNA. U, G, U, A, C, A, U, G, G, A, U, G, C, U, G, G, U, A. Now, there are 3 possible reading frames for an mRNA, okay? And that's due to the fact that there are 3 bases per codon, right? So, for example, this could be your open reading frame 1. So red is ORF1. We can have purple being the next reading frame, so starting there. So that's ORF2. And let's say green is the next one. Okay, ORF3. And that's all we can have, because look, if we then go to starting at the next one, this UGU, that's still the ORF1. Okay, so this is important. Okay, we have 3 different open reading frames. So if we search down here, we can find that there's an AUG. Okay, we have an AUG that we can find here and AUG that we find here. Those are start codons, right? And then we can find one UAA. This is a stop codon. Okay? So if we look, we can see that if we go red, 3, 3, 3, 3, 3, right? This is actually a start codon only for open reading frame 1. Okay? If we look for open reading frame 2, if we go 3, 3, 3, 3, 3, 3, 3, this is a stop codon only for open reading frame 2. And if we look over here, open reading frame 3, okay, this is 3, 3, 3. This is a start codon only for open reading frame 2. Oh, sorry. This should be in green. Okay, so this is important because let's say, for example, we're looking at our 3 possible open reading frames, right? So we see 2 different starts, okay, and 1 stop. Now, let's say, for example, that we're going to do our translation through open reading frame 3. So we can start here. We have AUG, that's going to be our start, our methionine. Then we'll have GAU as our next codon. Then GCU. Then GCU. Then UA, whatever that next one is. Okay, we'll write it in as, let's say, we'll just say A. Okay, now, all these starts and stops are perfect as they are now, right? But if we were to have a frame shift mutation, so either adding in or subtracting bases other than in 3, so you either add or subtract 1 or 2 bases, that can affect the open reading frame. So if we have an addition of a single base, okay, and if we say we add it right here, so a frame shift, okay, caused by the addition of an A right here, what that does is it shifts the open reading frame. So the new one, I'm gonna write it in red. Okay, so this would be AUG, GAU, GCU, GCA, UAA, A. Okay, so what we see here, now this is open reading frame 3. What we see is we have a start codon, an amino acid, an amino acid, an amino acid, a stop codon. Okay, so this is a truncated protein now. Instead of going on for maybe 500, 600 thousands of, amino acids, this now is a 4 amino acid polypeptide once it hits that stop codon. And likely this will just be sent to the cellular trash can, the proteasome, or the lysosome, and be broken down, degraded, and you would have to restart. Okay, so this is how a frame shift can affect the genetic code, because this might not be as big of a problem if we could overcome the frame shift, but if not, then maybe this protein isn't made at all in the body in its functional form, and that could possibly lead to a disease or cancer or something to where maybe it is a lethal type of mutation and there's not even life born from it. So that is the end of our unit today. Thank you for joining us at educator.com, and I hope to see you again next week.